Hello, my name is Sofia Hernandez, and I am the representative for my fellow peers, Jesus Espino, Isaac Rodarte, Michelle Palma, Edgar Ruiz, Ana Karen Garcia, and Carlos Araujo, and our mentors, Christopher Kuffner and Guadalupe Tapia. Today, we will be discussing the hydrolysis of PET by fusing modified hydrophobins to cutinase. So a little bit about PET. PET hydrolysis has been a major focus of study in enzymatic degradation of polyethylene terephthalate, commonly known as PET, and is one of the most widely used plastics in the world. It's commonly used in the clothing industry and the bottling industry. So various enzymes have been tested to degrade the highly crystallized PET, but have severely lacked in efficiency. And for this project, we chose to look into hydrophobins and a leaf branch compost cutinase, commonly known as LCC, an enzyme particularly suited toward these ends given its relatively high PET hydrolysis activity and thermostability. Hydrophobins are small cysteine-rich proteins commonly produced by fungus. They can naturally absorb to hydrophobic surfaces and form elastic films by self-assembling at interfaces, which could possibly create efforts for large-scale production. As we can see on the right in this figure, we can see the structural differences between two classes of hydrophobins. So class 1 has a diverse range of amino acids and has one varied intercysteine spacing. And some examples of these can include DU-A and HYD1, and class 1 can easily be manipulated, unlike class 2, which has a conserved amino acid sequence, has a short regular intercysteine spacing, and some examples include HFB4 and HFB7. So these hydrophobins do have some limitations. These limitations include inclusion bodies, which are a direct result of the formation of disulfide bridges between the cysteine residues among themselves. So we use a technique called protein fusion, which allows the fusion of LCC with another protein exhibiting hydrophobic properties and can significantly increase the hydrolytic capabilities. In one paper that we researched, it was a similar cutinase called THC cut1 was fused with a small selection of hydrophobins, and it improved hydrolysis 16-fold over the level with the free enzyme. We plan to express a fusion protein of LCC with the HYD1 class 1 hydrophobin in the bacterium E. Coli through the transformation using a plasmid prepared with general recombinant DNA techniques, followed by purification of the enzyme to use in PET hydrolysis to test how efficiently the PET is degraded for potential commercial use in other systems. On the left, we can see proteins fused by the protein linker and a model alignment of the LCC and THC cut1 that we researched. And here we have our experimental design, which we can see on the right. First, we started by preparing the plasmid for a fusion protein of LCC and the HYD1 hydrophobin. Then the E. coli is cultivated to be used for expression and is transformed with the plasmid. It's then incubated with various times, temperatures, and reagents, as we can see on figure 4. And then the bacteria is harvested, lysed, and centrifuged. So the hydrolysis is performed by incubating PET films with the lysate in buffer, and then the films are going to be washed and measured to determine the corresponding depolymerization rate. So to further advance this project, we would really need to be able to get together in a lab and fully test our experiment. Unfortunately, due to COVID and safety reasons, we could not do that, but we believe we did the most we could with what we had. But being able to get together would have made this project a lot easier. And we hope that next year's team incorporates elements of circular permutation to further improve the protein and the functionality of PET hydrolysis. We would like to extend our thanks to the following people who have helped us create and advance this project: Guadalupe Tapia, our teacher and mentor at school, and Christopher Kuffner, our mentor from Boston University for the BioBuilder Project Development Studio, who has provided advice and feedback on almost every angle of the project and helped with finding literature and developing ideas and software for this paper. We would also like to extend our thanks to Hope V, our assigned editor for BioChecks, and Dr. Natalie Codell, the founder and executive director of the BioBuilder Education Foundation, who has supported us through our presentations, media coverage, and research. Thank you for your time today. From the Silver BioBuilder team and I.